Welcome to another deep dive. Today we're going to be looking at a technique that's completely changed how we study cells.
Oh, wow.
It's called flow cytometry.
Okay.
Imagine a technology that can analyze thousands of cells every single second,
Yeah.
telling us their size, what's inside them, even what they're doing.
It's really incredible.
It's like having a super-powered microscope that gives us a real-time view of a hidden universe.
It really has revolutionized the field.
To guide us, we're diving into an article from Assay Genie
Okay.
called Data Analysis in Flow Cytometry, Harnessing Complexity for Scientific Insight.
Sounds interesting.
We'll be unpacking the science behind flow cytometry,
Mm-hmm.
the challenges of dealing with the data, and the real-world applications that are changing how we diagnose and treat diseases.
That's a lot to cover.
Yeah, but first, how does flow cytometry actually work? I mean, is it like Star Trek where they scan an alien life form?
Not exactly,
Yes,
but it does involve lasers. Yeah. Essentially, flow cytometry uses lasers to measure light scattering and fluorescence emitted by cells.
Okay.
Think of it like shining a flashlight through a glass of water
Okay.
with different sized beads in it.
Mm-hmm.
The way the light scatters
Yeah.
tells you something about the size and shape of those beads, right? Flow cytometry uses a similar principle.
So it's not just about taking a snapshot of a cell.
Right.
It's about analyzing how light interacts with it to reveal its secrets.
Exactly.
Like
We can
what?
take it a step further
How?
by tagging cells with fluorescent markers,
Oh.
like attaching tiny glowing beacons to specific molecules.
Wow.
When these markers are hit by the laser,
Yeah.
they emit light at particular wavelengths,
Okay.
allowing us to identify and track those molecules within the cell.
So we're not just seeing the cell's external features.
Yeah.
We're actually peeking inside.
Yeah.
Like seeing the compartments and molecules.
That's right.
But hold on.
Yeah.
If we're analyzing thousands of cells per second, each with all these different markers,
Yeah,
that must generate an insane amount of data.
it does.
I mean, how do scientists even begin to make sense of all that?
That's a great question.
Yeah.
You've hit on a major challenge in flow cytometry,
Okay.
the sheer volume of data.
Right.
It's like trying to understand the workings of a city
Okay.
by monitoring the movement of every single person, every car, every bus.
Whoa.
You need powerful tools to filter, organize, and interpret that information.
That's where the data analysis part of the article title comes in, right?
Exactly.
What are some of the key techniques that help scientists unlock these insights hidden within this data deluge?
One of the most fundamental techniques is called gating.
Okay.
It's a bit like sorting laundry.
Oh, it's like separating clothes.
Exactly.
Okay.
By color fabric type,
Mhm.
or what they're used for.
Mhm.
In flow cytometry, researchers use gates to select specific cell populations based on their characteristics.
So if you're interested in, say, T cells,
Yeah.
which are a key part of our immune system, you could set up a gate that singles them out from all the other cells in a blood sample.
Precisely.
That makes a lot of sense.
It allows researchers to focus their analysis on specific cells,
Right.
making the data much more manageable.
That makes sense. But remember,
Yeah.
we're often dealing with complex relationships between cells,
Right.
not just individual populations.
It's true.
It's not enough to just identify the players. We need to understand how they interact with each other, like a cellular social network.
Exactly.
How do scientists go beyond simple sorting to uncover those hidden relationships?
That's where things get even more interesting.
Okay.
We use a range of tools,
Okay.
including statistical methods like principal component analysis and cluster analysis.
Wow, those sound complicated.
They can be, but they help us reveal hidden patterns and correlations within the data.
Okay.
Imagine you're at a large gathering,
Okay.
and you want to figure out who knows
Mm-hmm.
who hangs out with whom.
Right.
These tools can help us find those clusters of cells
Uh-huh.
that share certain characteristics or behave in similar ways.
So we're going beyond simply categorizing cells
Yes.
and starting to see the bigger picture, like understanding how different cell types work together to
Exactly.
keep our bodies functioning,
That's right.
or how they might go awry in a disease.
Exactly. This is where flow cytometry really shines,
Okay.
allowing us to see beyond the individual cell and understand how these complex systems interact.
I see.
But even with these techniques, things can get tricky.
How so?
When we're dealing with data that has dozens of parameters for each cell,
Wow.
it's like trying to visualize a multidimensional object. We need specialized tools for that.
What kind of tools?
One example is t-SNE.
What's that?
It stands for T-Distributed Stochastic Neighbor Embedding.
Okay.
It's a mouthful, I know.
Yeah.
But what it does is take this complex, high-dimensional data and reduce it to a lower dimensional space that we can actually visualize.
So we can actually see it.
Yes. Think of it like creating a simplified map that highlights the most important landmarks and routes.
Oh, so it's like taking a really complex map
Yeah.
with layers upon layers of information
Right.
and creating a more user-friendly version that still captures the essential relationships.
Exactly. These tools allow us to see the forest for the trees.
And revealing patterns and connections that would be incredibly difficult to discern in the original data.
That's right.
And those patterns can hold the key to understanding disease processes
Exactly.
or identifying new drug targets.
Yes.
This is all incredibly fascinating.
It is.
But I'm also wondering about the practical implications.
Okay.
How is this knowledge actually being used to make a difference in the world?
It's used in many fields,
Really?
like immunology, oncology, and stem cell research.
Oh, wow.
For example, in immunology flow, cytometry is helping us understand how the immune system responds to infections or vaccines.
Oh, interesting.
We can track specific cell types like those T cells you mentioned earlier.
Yeah.
And see how their numbers and activity change over time as the body fights off an invader.
So it's like having a front row seat to the immune system's battle strategies.
Exactly.
Wow. And in oncology, I imagine flow cytometry plays a key role in understanding and treating cancer.
Absolutely.
How so?
It's used for a wide range of applications,
Okay.
from diagnosing and classifying blood cancers to monitoring how well treatments are working,
Mm-hmm.
and even detecting minimal residual disease.
Minimal residual disease. What is that?
It refers to the presence of a small number of cancerous cells that can remain,
Oh, wow.
even after treatment appears to be successful.
Really?
Yes.
So they're hiding.
Yes, these cells are often undetectable with traditional methods,
Yeah.
but can lead to a relapse.
If not addressed.
Exactly.
Oh, wow. So flow cytometry can actually see these cells.
Yes, flow cytometry's ability to detect and analyze these rare cells is critical.
For guiding treatment.
Yes, and improving patient outcomes.
It's amazing how this technology is not only helping us understand cancer better, but also leading to more personalized treatment strategies.
Absolutely.
What about stem cell research? How does flow cytometry contribute to that field?
Stem cell research relies heavily on flow cytometry for identifying and isolating different types of stem cells,
Okay.
each with unique characteristics and potential for developing into various cell types.
So flow cytometry is helping us unlock the potential of these remarkable cells
That's right.
for regenerative medicine and potentially treating a wide range of diseases.
Exactly.
This is all incredibly exciting.
It is.
But before we go further, can you remind us again,
Okay.
in simple terms, what's happening at the heart of a flow cytometry experiment?
Well, imagine a single file line of cells
Right.
being rapidly transported through a tiny channel, almost like a cellular conveyor belt.
Mm-hmm.
As each cell passes through a laser beam,
Yeah.
it scatters light.
Okay.
And if the cell is tagged with those fluorescent markers we talked about, it also emits light at specific wavelengths.
So we're essentially using light to interrogate each cell as it zooms by,
Yes.
gathering information about its size, its internal contents, and whatever markers we've attached to it.
That's a great way to put it.
It's like a high-tech cellular interview.
Exactly.
And these signals from the scattered and emitted light are picked up by detectors,
That's right.
which convert them into digital data that
Yeah.
we can then analyze.
Exactly.
And remember, we're analyzing thousands of cells per second.
That's right.
So we're getting a massive amount of data very quickly.
We are.
It's amazing how much information we can glean from something as simple as light.
It is.
It's not just about seeing the cells.
Right.
It's about understanding their individual stories and how they contribute to the larger narrative of the organism.
Exactly.
And that's what makes flow cytometry so powerful.
It is.
We're not just looking at a few cells under a microscope.
Right.
We're getting a comprehensive view of entire cell populations and their incredible diversity,
It's like having a census of the cellular world.
revealing not just the numbers,
Yeah.
but the unique characteristics of each individual.
That's a great way to put it.
And with that, I think it's time to move on.
Okay.
We'll delve deeper into the data analysis side of things, exploring the challenges and the triumphs of deciphering the language of cells.
Sounds good.
Stay tuned.
Okay.
Welcome back. Before the break, we were talking about how flow cytometry is like conducting a high-tech cellular interview
Right.
using light to uncover the secrets of individual cells.
Exactly.
It's mind-blowing how much information we can gather about each cell's size, internal components, and even its function.
It is remarkable, isn't it?
Absolutely. We touched on this earlier, but I'd
Yeah.
love to unpack those data analysis techniques a bit more.
Okay.
You mentioned gating
Right.
is one of the foundational techniques.
Yes, one of the most fundamental.
It sounds straightforward enough,
Mm.
separating cells into different groups
Right.
based on their characteristics. But I imagine it can get quite complex in practice,
It can.
especially with all the different parameters we can measure.
You're right, gating can be quite intricate.
Yeah.
The concept is simple.
Okay.
Think of it like setting filters
Okay.
to choose specific cells, but the execution
Yeah.
can become very sophisticated.
How so?
Researchers might use a series of gates,
Okay.
each one refining the selection further
Oh, wow.
to isolate very specific cell subsets.
So it's not just a single filter.
No.
It's more like a multi-step process
Yes,
of
sir.
refinement, kind of like sifting flour multiple times to get the finest texture.
That's a great analogy.
And these gates can be based on a variety of parameters,
Yes.
not just the presence or absence of a specific marker.
That's right.
We can use gates to select cells based on size, internal complexity, or even a unique combination of markers.
Exactly.
So this allows researchers to really pinpoint those cells that are most relevant to their research.
Precisely.
It's like finding a needle in a haystack.
Right.
But instead of searching randomly, we have a set of tools that help us zero in on the exact needle we're looking for.
Exactly. And thankfully,
Yeah.
there are software programs that help researchers visualize the data and set up these gates in a more intuitive way.
So we're not just dealing with spreadsheets and numbers here. We're
Haven't-
actually visualizing the data,
That's right.
seeing those clusters of cells emerge based on their characteristics.
Exactly.
That must make the process a lot more manageable.
It does. These software programs allow researchers to create two-dimensional plots
Okay.
where each dot represents a single cell,
Okay.
and its position on the plot reflects its values for two chosen parameters.
So like a graph?
Kind of, yeah.
Okay.
For example, one axis might represent cell size,
Uh-huh.
and the other could show the level of a specific protein.
It sounds almost like creating a map of the cellular landscape,
That's a great way to think about it.
with different regions representing different cell populations.
Yes, and by drawing gates around specific regions on these plots, researchers can isolate the corresponding cell populations for further analysis.
But even with these visual tools, gating can sometimes be subjective,
It can be.
especially when the cell populations are not clearly separated.
Right.
So there's still an element of human judgment involved,
There is.
which could potentially introduce variability or bias into the analysis.
That's the valid point, and that's where automated gating algorithms come into play.
Automated gating algorithms.
Yeah.
So we're bringing in some artificial intelligence to assist with the cell sorting process.
Precisely. Automated gating algorithms use statistical methods and machine learning
Wow.
to identify cell populations in a more objective and reproducible way.
So it takes out some of the guesswork.
Yes, they can analyze the data and suggest optimal gate placements
Okay.
based on the underlying patterns, reducing the potential for human error or bias.
That sounds incredibly powerful, but also a bit complex.
It can be.
Can you give us a simple example of how these algorithms might work?
Imagine you have a plot with thousands of dots representing cells,
Okay.
and you want to group them based on their size and granularity.
Okay.
A clustering algorithm could analyze the distances between the dots on the plot and identify groups of cells that are close together based on those two parameters.
So it's like the algorithm is drawing circles around groups of cells
Yeah.
that share similar characteristics,
Exactly.
kind of like grouping stars into constellations
Right.
based on their proximity in the night sky.
Exactly. These clusters identified by the algorithm can then be used to define gates and isolate the corresponding cell populations.
And as these algorithms become more sophisticated,
Yeah.
they're playing an increasingly important role in analyzing the massive data sets generated by high-parameter flow cytometry.
They are becoming essential tools,
Right.
especially as we move towards analyzing more and more parameters per cell.
This brings us back to something you mentioned earlier, high-parameter flow cytometry.
Yes.
We're increasing the resolution of our cellular microscope.
Right.
But that also means we need even more powerful tools to make sense of the picture.
Exactly. And this is where multivariate analysis techniques become crucial.
We talked about multivariate analysis briefly,
You did.
but can you remind us why it's so important for analyzing flow cytometry data?
Of course. Multivariate analysis allows us to explore the relationships between multiple parameters simultaneously,
Okay.
revealing patterns and correlations that we might miss if we only looked at each parameter in isolation.
So instead of looking at individual pieces of a puzzle, we can see the whole picture.
Precisely. One common multivariate analysis technique used in flow cytometry is principal component analysis,
Okay.
or PCA.
TCA.
Yes, imagine you have a data set with measurements of 10 different parameters for each cell.
Okay, 10 different parameters.
PCA can take those 10 dimensions
Okay.
and compress them into a smaller number of dimensions,
Oh, wow.
say 2 or 3,
Okay.
while still capturing as much of the variation in the data as possible.
So instead of getting lost in a 10-dimensional data jungle,
Right.
we're creating a simpler map that highlights the most important features.
That's a great way to put it. These new dimensions, called principal components,
Principal component.
are essentially weighted combinations of the original parameters,
Okay.
chosen to maximize the variance explained by each component.
Okay.
By plotting the cells in this new lower dimensional space,
Mhm.
we can often see patterns and relationships
Yeah.
that were hidden in the original data.
It's like using a special filter to highlight the most relevant information,
Yes.
making it easier to spot those hidden connections.
And there are many other multivariate analysis techniques that can be applied to flow cytometry data,
Oh, wow.
each with its own strengths and weaknesses.
Okay.
But the key takeaway
Yeah.
is that these techniques allow us to go beyond simple gating
Okay.
and delve into the complex relationships between different cell types and their characteristics.
This is all incredibly fascinating.
It is.
But I have to admit,
It can feel a bit overwhelming at times.
I understand.
It's like learning a whole new language.
It is a lot of information.
The language of cells.
Flow cytometry data analysis can be quite complex,
Yeah.
but it's also incredibly rewarding.
Okay.
By mastering these tools, researchers are gaining unprecedented insights into how cells work,
Right.
leading to groundbreaking discoveries in various fields.
It's amazing to think that we can learn so much
It is.
from something as simple as shining a laser beam at a cell.
It's like unlocking a hidden universe within each tiny cell.
It truly is.
And as technology continues to advance, I have no doubt that flow cytometry will continue to push the boundaries of our understanding of life itself.
Well, I think we've covered a lot of ground today.
We have.
From the basics of flow cytometry to the complexities of data analysis.
Right.
But before we wrap up this deep dive, I'm curious to hear your thoughts on what the future holds for this incredible technology.
Okay.
What exciting developments are on the horizon? Well, it feels like we've only just scratched the surface.
Where do you see flow cytometry heading in the future?
One of the most exciting areas is high-parameter flow cytometry,
Okay.
where we can measure dozens, even hundreds of parameters on a single cell
Hundreds.
simultaneously.
Wow.
It's like having a microscope that can not only magnify an image, but also reveal the chemical composition,
Okay.
the genetic activity, and countless other features,
All at the same time?
all at the same time.
That's incredible, but how do you analyze all that?
It definitely requires specialized tools and expertise.
Right.
But the beauty is that the field of data analysis is also evolving rapidly,
Oh, okay.
with advancements in machine learning and artificial intelligence providing powerful new approaches to handle this complexity.
So these algorithms
Yes.
can sift through massive data sets,
Right.
identify patterns,
Yes.
and even predict how cells might behave under different conditions.
Exactly. It's like having a team of super sleuths working behind the scenes,
Right.
uncovering hidden connections and revealing insights that would be impossible for humans to see on their own.
And the potential applications of this are vast.
Oh, they are vast.
Can you give us an example?
Imagine using high-parameter flow cytometry to identify a unique set of markers
Okay.
on cancer cells.
Okay.
That could predict how well a patient might respond to a specific treatment.
So personalized medicine,
Exactly. This could help doctors tailor therapies to each individual.
leading to more effective treatment.
Precisely.
Wow, that's amazing.
And this is just the beginning.
Really?
Another exciting direction is the integration of flow cytometry with other technologies,
Oh, like what?
like imaging and genomics.
Oh, okay.
Imagine combining the detailed cellular analysis of flow cytometry with the spatial information provided by imaging.
We can actually see where the cells are.
Yes, we could see exactly where specific cell types are located within tissues and how they interact with their neighbors.
So it's like adding a whole new layer of understanding to the cellular landscape. Wow, and what about genomics?
By linking flow cytometry data with genomics,
Yeah.
we can connect those cellular phenotypes to the underlying gene expression patterns.
So we can see the genes that are being expressed.
Exactly.
That's incredible.
This allows us to understand not just what a cell looks like or what it's doing,
Right.
but also what genetic programs were driving those characteristics.
It's like getting a glimpse into the instruction manual of a cell.
That's a great analogy.
Yeah.
And as these technologies continue to converge,
Yeah.
we're only going to gain a deeper and more nuanced understanding of the complexities of cellular life.
It's clear that flow cytometry is a transformative technology.
It is.
With a bright future. Any final words of wisdom for our listeners?
I encourage everyone to check out the Assay Genie article we've been discussing.
Yeah, it's a great article.
It's a great starting point for understanding the fundamentals of flow cytometry and its diverse applications.
Right.
From there are countless resources available. Research articles, online courses, even conferences dedicated to flow cytometry,
Wow.
where you can connect with experts in the field.
It's a field that's constantly evolving.
It is.
With new discoveries being made all the time.
That's right.
Who knows what incredible breakthroughs await us in the years to come.
The possibilities are truly endless.
That's exciting.
Whether you're a student, a researcher, or simply a curious individual exploring the world of flow cytometry is a journey well worth taking.
I completely agree. Well, thanks for joining us on this deep dive into the captivating world of flow cytometry. We hope you've gained a new appreciation for the power of this technology and the incredible insights it's providing into the mysteries of life itself.